Please go ahead, and we are looking forward to the presentation of Alexandros.
Yeah, thank you very much, and thanks the organizers for such a nice session. And it's unfortunately very sad that two of our speakers are not here, but we hope next year things will be better. Nevertheless, I'm here today to describe you one of our research branches that we do. We are focusing on using nanomedicines for targeting immune cells. And this particular branch, we are naming it Hitchhiking. And what you see in the first image here is an image that I generated with Midjourney AI. So I typed nanomedicine hitchhiking for tumor implosion, and that's the result I took. And if you see here at this blue bubble, it's a very blurry thing that is supposed to be the Cathedral of Aachen. So that was the AI thought about it, and give me this tiny little thing that you need a magnifying lens to project. So what we are speaking about when we talk about nanomedicine, so we have vessels, we inject intravenously our nanoparticles, which they circulate, and at some point they target specific myeloid immune cells. They can either target these immune cells on their receptor, and they can be taken up, or they directly can be phagocytosed. And progressively, those immune cells, either in cancer or inflammation or other models, they have the tendency to extravasate. So they are localizing themselves, targeting the malignant lesions. Progressively, a lot of them, they go out and they are actively delivering nanomedicines in the microenvironment. So we started this research several years ago. We were focusing on nanoparticles decorated with CRGD peptide. It's a very common peptide that's supposed to target the alpha V beta 3 integrin. Our first study was focusing on designing these nanoparticles and see how they can target tumor. Before that, we were very interested to see how they behave in vivo. And for that, we used different imaging modalities, multi-scale imaging at different spatiotemporal resolutions. Started with PET, continued with intravital microscopy, and also corroborating those evidences by ex vivo analysis. And what we saw by intravital microscopy that you can see on the left, that as soon as we inject these RGD nanoparticles in circulation, we see that some nanoparticles, this blurry thing in the vessels, are floating nanoparticles, but you can see some very dense speckles in the middle. So these are nanoparticles that have been taken up by immune cells. And if you see that some of those immune cells, they are exiting from the vessels, carrying the loaded nanoparticles into the tumor microenvironment. And if you see on the right side a time frame, so as soon as this immune cell has taken up nanomaterial and has stuck on the endothelium, the entire process of extravasation takes approximately 8 minutes. It seems that those immune cells, they are very motile. They are first capable of taking up nanomaterial, and second, they are able to very fast extravasate. And we thought that flow cytometry would solve the question which immune cells are interacting with those nanoparticles. And we very convincingly found that neutrophils are responsible with engaging with RGD nanoparticles and at multiple time points delivering them to the tumor. Now, focusing on intravital microscopy and specifically on neutrophils, you can see here the tumor microenvironment in which we have stained neutrophils with blue. You can see, I play the video, they go back and forth. This is real time with very fast temporal resolution. And now, in the same mouse, the exact same field of view, we have also injected control nanoparticles without the RGD decoration. And if I play this, you see that these nanoparticles are circulating in the vessels. This is 4 hours post-injection. On the bottom left, you can see some of these nanoparticles, they have extravasated and they have captured by tumor-associated macrophages. Now, for the very same time point, if I display to you the co-injected RGD nanoparticles, you see a completely different profile. So you see that we don't have them in the circulation anymore. Instead, we have in balls of fluorescence that they're moving back and forth. And if I play everything together, and you focus on the zoom on the left, you can see a lot of colocalization between blue neutrophils and red that they are the RGD particles. So we have a very aggressive interaction between the two. I can tell you that the positivity in blood can be over 99% for certain time points. Then, why do neutrophils engage so much with RGD nanoparticles? Is because they are co-expressing the alpha V beta 3 integrin for which the RGD peptide is meant to target. Then following our observation in cancer, we wanted to use a second model, and this model is on inflammation. We wanted to see if similar results we could identify beyond cancer. In this case, we use an inflammation model. We inflicted an ear cut in the mouse that you can see in the picture, and then we continued with intravital microscopy again. You can very briefly see here the difference. So on the left, the RGD nanoparticles, on the right, the control nanoparticles that co-injected, and you see inside the immune cells only the first case, which we later corroborated by ex vivo analysis, ex vivo microscopy. So we took out blood and we could see RGD nanoparticles taken up a lot by neutrophils, and we quantified this to be at least 1 or even 2 orders of magnitude higher in comparison to what happens to control particles. And in the next model, so the first 2 models we use, a lot of immune cells, they are responsible for having a role in the tumor microenvironment. But now we use the different model, sepsis, because in sepsis, neutrophils is the first player that aggressively migrates into the inflamed, into the septic area to resolve the disease. So the model we used is a cecal ligation and puncture. So we inflict a small cut in the abdomen, we take out the gut, we tie it, and then we puncture it such that we initiate the septic process. So if we leave the mice untreated, after 1 day they die. So this is a very aggressive acute inflammation model. And as such, since it's so aggressive, other phenomena that we have in cancer, for example, angiogenesis, they don't have time to occur. So this would be a very nice model to really see the contribution of neutrophils in delivering material. Indeed, we did first whole body imaging, specifically fluorescent tomography corroborated by computed tomography. And you can see over time that we have targeting both in the liver but also a lot in the intestine, particularly the side in which we had inflicted the CLP model. And you can see at the bottom the ex vivo analysis that in the SAM model, where we did not initiated the model, we have no targeting, but in the right side we have quite some targeted in the area. And if you see the analysis on the right, between control liposomes and RGD liposomes, we see that in the intestine, the dominant population, the dominant nanoparticle that targets the lesion are RGD liposomes. Why they are RGD liposomes? Because of neutrophil contribution again, right? They are massively infiltrating. You can see on the right side that the RGD uptake by neutrophils is very aggressive. On the left side, you can see monocytes and macrophages. That is, if you can appreciate the SAM model where we don't have the actual development of sepsis, the uptake by monocytes and macrophages is higher because this is what macrophages do. They are phagocytosing. And then when we have the CLP model, we initially see a very strong transition of the dose from other populations to neutrophils that eventually infiltrating into the septic side. We have moved this 1 step forward, and this is the first proof that hitchhiking can be considered for therapeutic applications. We have loaded liposomes and RGD liposomes with dexamethasone, and we see that we can even tone down furthermore the inflammatory effect by using RGD liposomes. And at this point, after doing a lot of validation in different models that hitchhiking can occur in different diseases and can potentiate active targeting, we wanted to see how much those neutrophils contribute to active targeting. So out of 100 nanoparticles that reach the tumor, how many do it with other mechanisms and how many do it via hitchhiking with neutrophils? And to do this, we have a big cohort of mice. Half of the cohort we injected PBS, so control group, but in the other one we injected the monoclonal antibody that depletes neutrophils. And after 48 hours, which is a very important time point, because at 48 hours all neutrophils are gone, but the tumor microenvironment has not reshaped. So it's a very sweet spot to identify what is the actual contribution of neutrophils into targeting the tumor. And then we injected, of course, control and RGD liposomes, and we tracked them over 1 day at different time points with fluorescent and computed tomography. I would like you to focus in the H and T for heart and tumor. So by quantifying the activity in the heart, we can see the circulation half-life of the liposomes, and by looking at the tumor, we can see the targeting into the tumor. So first of all, if we see the hard for control liposomes, you see that when neutrophils are depleted, the circulation half-life slightly increases. The reason for that is that neutrophils are slightly, of course, they are phagocytic, not that much as macrophages. They are not clearing particles as fast as macrophages do, but they are improving slightly circulation half-life. And this also reflects into the tumor targeting. So the fact that liposomes remain a little bit more in circulation allows them to target tumor a little bit more. However, in the quantification, you see that these differences are just minor. When we go now to RGD liposomes, you see again in circulation that the lack of neutrophils helps them to remain in the blood a little bit more. But if you now focus on tumor targeting, you see that we have a huge drop in the targeting for RGD particles because there are not neutrophils anymore into the circulation to drive them there. And the quantification, we have calculated a significant drop of 65 to 70% if neutrophils are not in the game. And to show you a visual flare about it, on the left side, you see how liposomes are targeting the tumor with and without neutrophils, so more or less same magnitudes of targeting. But you can appreciate now when we have RGD liposomes that in case of neutrophil depletion, we have a very huge drop in the tumor targeting. So far we have only spoken about engagement between RGD particles and neutrophils. But as you may know, the RGD peptide for targeting the alpha V beta 3 integrin has traditionally been used also for targeting the angiogenic endothelium. For example, a very typical experiment that we do even in early career stage is to have HUVEC cells, activate them, they express alpha V beta 3, and then we inject RGD nanoparticles in the system and see how they engage in vitro. So then we wanted to see what is happening as a bonus insight, what is happening in vivo with these. So you see here when we isolated from the tumor microenvironment CD31 positive endothelial cells, that for control and RGD particles, at 4 and 24-hour time points, we see exact same targeting. So endothelial cells pretty much, they are taking up the different particles at the same levels. And for 12 hours even, you can see that control nanoparticles are taken up more because they are just following the circulation half-life profile. Control passive targeting nanomedicines, they are typically more stealth, so they are hiding the cell from the immune system, and as such, they are cleared slower. In case of RGD nanoparticles, they are of course cleared faster because of multiple reasons. And because of that, the active targeting towards endothelium, it doesn't work in vivo. And to investigate this even further, we did a study on cellular competition. So if we have at the same time neutrophils and endothelial cells in the environment, how would they differently engage with RGD nanoparticles? So first, let's focus on neutrophils. On the top, you can see neutrophils in green, RGD nanoparticles in red. You can see quite some colocalization between them, and which is approximately 1.2% of the area fraction. At the bottom image, you can see the microenvironment without neutrophils when the neutrophils are depleted. So you don't see obviously green, you see only particles, and obviously the colocalization between the two is 0. Now, if we focus on the endothelial cells, now on the right-hand side, you can see the endothelial cells in green. You see that they have a completely different spatial distribution that RGD nanoparticles. They don't engage with them. And as you can see with the dark color here, the RGD nanoparticles colocalization with endothelium is very, very low when neutrophils are in the system. But in this image that we have depleted neutrophils, you start seeing colocalization between RGD liposomes and endothelium. And you can see how much more we increase their engagement. So this means that the concept of using integrin-targeted nanoparticles for angiogenic reasons would work only if phagocytes are not anymore in circulation or in the tumor microenvironment. So this brings us a realization that all active targeting nanomedicines that we may have used as community in the past, maybe they would be very efficient and would not fail clinical trials if we had the right target for them. So to come back and conclude, What we saw is we designed different type of nanoparticles and we decorated them with RGD. We did liposomes, nanoemulsions, PEG-PLGA polymers, different sizes. We initially injected them in a tumor mouse model, 41 and derivative of those. We have done this in 3 models. We injected them intravenously. And what we saw in circulation is that the RGD liposomes, they do not target directly the endothelium. They do not extravasate, but instead they are taken up by neutrophils, which they progressively infiltrate into the tumor microenvironment, actively carrying the dose in it. And we certainly followed up by having observation in wound healing, also in sepsis, and we calculated this contribution to be around 70%. And as of now, we are trying to move this concept into therapy. So we are working to see what kind of nanoparticles could also facilitate neutrophil-mediated transportation in the tumor, and how we can further enhance chemically or biochemically neutrophil infiltration in malignant lesions, and last but not least, to see how this engagement, what kind of cargo would allow us to carry, small molecules, nucleotide therapeutics, and so on. And by this, I would like to thank a lot, first of all, Sjurd Hack from NTNU and SINTEF from Norway. We started these projects together 7 years ago, and Professor Toine Lammers from Aachen. We do quite a lot of cool studies on immune cell targeting these days. My team in Aachen, and of course, collaborators, institutes, and funding agencies. And thank you very much for your attention.
Thank you very much. Thank you very much, Alexandros. Very good presentation.